हाँ धर्मेंद्र जी। ओके ओके। स्टार्ट तो किया ही हुआ है। हम्म। ओके ओके ठीक है। ओके okay. So sorry, there was some technical glitch. So let us begin with the topic. So why this topic is important? Three parent baby and therapeutic cloning. So three parent baby, it is important because it was in news. So this article had come in Hindu and it was covered in almost all newspapers at that point of time. So it has been in news. So in fact, it has been in news from past many years. So the three parent baby, this had has been made legal in UK few years back, but it has been made legal even in your Australia. So that is why it is in news. So UPSC has already asked questions based on this on mitochondrial replacement therapy or three parent baby. So what are we going to do? We are trying. We'll develop an understanding of what is this three parent baby, and then we'll also see this therapeutic cloning. So these are some of the applications of biotechnology. Now, biotechnology overall has become a very important topic, especially after COVID. So you would have seen questions being asked in prelims, questions being asked in mains. So in prelims, they've asked questions on your understanding of vaccines, your understanding of microbes like bacteria and fungi, even algae. And they have in mains they've asked questions based on your understanding of vaccines. What is the principle of the vaccine? How does vaccine work? So biotechnology has become important. So in biotechnology, one of the key application is in what we call as the manipulation of the cell itself. So in order to understand mitochondrial replacement therapy, we need to understand that why one has to go for mitochondrial replacement therapy. So this involves the understanding of DNA itself. So DNA is called as what? DNA is the mechanism through which your inheritance is passed on from one generation to another generation. So what we generally understand that DNA, or this deoxyribose nucleic acid, which is the carrier of the genetic information, it is mainly stored in your nucleus. So 99% of the DNA is present inside nucleus, but 1% of the DNA is also present inside mitochondria. So what you are seeing here is a typical animal cell. So human cell is also very similar to this only. So in human cells also you will find that you have this nucleus. So let me just mark it. So you would be aware that this is an animal cell, and in animal cell you would have typically a nucleus at the center of the cell, and then you would see these thread-like structures. These thread-like structures inside the nucleus. So these thread-like structures inside the nucleus, they are basically your chromosomes. And if you open up the chromosomes, what you will see that the chromosome is made up of your some functional units or some parts which are called as your DNA. So your chromosome is nothing but your DNA. So if you add more and more DNA, you basically build up chromosome. So chromosome is essentially a packaged DNA. So this DNA carries the information regarding our characteristics, our traits. It carries information for some of the diseases also. So this DNA, what we generally believe, is present mostly inside nucleus, but in animal cells, in humans, you'll see that some amount of DNA is present outside the nucleus also. So some of the DNA is present in the mitochondria. So you would have read in school, or you would have remembered from school, that mitochondria is the powerhouse of the cell. But here, along with that function, it also carries some DNA. Now, if there is some defect in this type of DNA that is present inside your mitochondria, then it can be passed on from the parents to the child. And there is a unique characteristic of this mitochondrial DNA. The unique characteristic of this mitochondrial DNA in humans is that the mitochondrial DNA is passed only from the mother to the child. That is the uniqueness of the mitochondrial DNA. So if mother's mitochondrial DNA is defective, then there is a high probability that the child will also inherit the defective mitochondria. So in order to get rid of this situation or this problem, we have to go for mitochondrial replacement therapy. So in UK it has been legalized, in Australia it has been legalized, but in other countries they are still working out that what has to be done with this type of therapy. So if a person inherits this mitochondrial defective mitochondrial DNA, then he or she is prone to some of the diseases. So what this slide is saying, that this can cause something which is referred as the Lay syndrome. So Lay syndrome is a neurological disorder. So a child which carries defective mitochondrial DNA might have this Lay syndrome. So he or she could experience this severe disease. So it also says that faulty mitochondria have also been linked to more common medical problems, including Parkinson's disease, deafness, failing eyesight, epilepsy, and diabetes. So these are some of the medical concerns related to the mitochondria. So the defective mitochondria, if it is passed on to the child is vulnerable to such phenomena such as your Lay syndrome, Parkinson's disease, or your deafness, falling eyesight, epilepsy, and diabetes. So what is the cure for this? So the medical cure or the treatment that we have found is basically we replace the defective mitochondria. So how do we replace the defective mitochondria? So there are basically two methods of replacing the defective mitochondria. So one of the method is referred as, so there are two methods to replace mitochondria. So what we are seeing is mitochondrial replacement therapy. Your mitochondrial replacement therapy, we also, or this is popularly referred as three parent baby. We'll see why we are calling it as a three parent baby. So mitochondrial replacement therapy is popularly known as three parent baby. So why we are calling it as three parent baby? Because we are going for in vitro fertilization. What is in vitro fertilization? Fertilization is the process in which the egg cell and the sperm cell are fused together to form a zygote. So during this fertilization process, we can do some manipulation, some genetic manipulation. So this is what is referred as your mitochondrial replacement therapy. So there are two methods of doing this mitochondrial replacement therapy. First method is called as maternal spindle transfer. We'll see how it is done. So first method is called as maternal spindle transfer, and a second one is called as pronuclear transfer method. So UPSC has already asked questions based on your understanding of this method and pronuclear method, also pronuclear transfer method. So sometimes they ask the scientific name itself, or they could ask the technique that is being used in this pronuclear transfer method. So what we do in the maternal spindle transfer method. So in the maternal spindle transfer method, the idea is that if the mother's egg cell is defective, it means it's cytoplasm. So see, the diagram might be crude, but this is just to develop our understanding. Let us say this is mother's egg cell. This is the mother's egg cell, and in the mitochondria, these red crosses that I'm drawing, these are your defective mitochondria. These are your defective mitochondria or defective mitochondrial DNA. So what we are trying to do here is that we will try to get rid of the defective mitochondrial DNA. So we have some techniques. So what we can do, this is your nucleus. So the nucleus is okay. So in the mother's egg cell, this is the mother's egg cell, but her egg cell is having defective mitochondrial DNA. And what we are going to do is to replace these defective mitochondrial DNA with someone else's nucleus, so with someone else's cytoplasm. So this area outside your nucleus is called as your cytoplasm. So let us say this is the initiating mother, this is the initiating mother's egg cell, this is the initiating mother's egg cell. So what we'll do, we know that her cytoplasm is containing defective mitochondria, so we have to get rid of this cytoplasm. So what we do, we can remove the nucleus in this process, and this process is known as enucleation. Enucleation. So what we are doing, we know that the nucleus is okay or nucleus is healthy, the cytoplasm is containing the defective mitochondria. You must also take a note that the number of mitochondria in in an egg cell is quite a lot. So mother's mitochondria or mitochondrial DNA, so there would be around more than 2 lakh mitochondria. So we cannot replace and pick and choose or remove the defective ones. So what we do instead is that we get rid of the cytoplasm, complete cytoplasm itself. So we extract the nucleus. We know that the nucleus is okay, the nuclear DNA is okay. So we'll remove this nucleus and we'll leave behind the cytoplasm. So this cytoplasm is containing the defective mitochondria. So we have got the healthy nucleus, but we also need what a cytoplasm which contains, let me use just a different color. So this is a donor mother's egg cell. This is donor mother's egg cell, and this donor mother's egg cell, her egg cell is containing what? Healthy mitochondria. So she is having healthy mitochondrial DNA, but we do not want her nucleus or her genetic information being passed on to the child, because the offspring that is going to be born is basically a child of, should be a child of this mother, should be a child of this mother. So what we are doing, we will get rid of this nucleus. So again we'll go for enucleation, because we do not want the nucleus of the donor mother, we just want the cytoplasm of this donor mother. So the idea is that we'll take the healthy nucleus from the initiating mother or the original mother, and we'll get rid of the defective cytoplasm which is containing the defective mitochondria. Now what we are doing parallelly is that we are taking a donor mother's egg cell and we are removing the nucleus, because we do not need her nucleus. We do not want that her donor mother's genetic information should be passed on to the child. We only want her healthy mitochondria. So we'll extract or we'll remove this nucleus. Now we have got the healthy cytoplasm. This cytoplasm is containing the healthy mitochondria. So now we'll fuse them together. So this and this we are fusing it together. So I'm using a pen tab, so diagram looks little crude, but this is for your understanding only. Now this, what you're seeing, that we have got the nucleus from the original mother, we have got the nucleus from the original mother, and we have got the cytoplasm from the donor mother. Now this is a new egg cell. This will behave as a new egg cell, and this new egg cell is containing the nuclear DNA of this mother, and it is containing the mitochondrial DNA of this mother. So this new egg cell is basically composed, or it is carrying genetic information from, mostly from the initiating mother, but it is also containing some or 1% of the information, genetic information, from the donor mother also. Now what do we do? Now we have got a egg cell, and we'll go for IVF. IVF is what? In vitro fertilization. So in vitro fertilization means, so there are two types of fertilization. Fertilization is basically when the egg cell and the sperm cell, they fuse to form a zygote, the first cell from which we develop. So the first cell that is formed, that is called as your zygote. So in this case, we are going to go for IVF only after the new egg cell has been formed. So we will take this egg cell, and what we'll do, we will fertilize it with a sperm cell. So let us say this is a sperm cell, and this also has a nucleus. So this is a sperm cell, and both of them will be fertilized. So we will get now what we will get. So what happens when we try to fertilize is that the nucleuses from both the mother side and from the father side, they just sit side by side for a very short period of time. So they do not fuse instantaneously, but they take some time, and this stage is called as your pronuclear stage. Pronuclear means before nucleus, before one nucleus. And after this, what happens, that okay, so now this pronucleus is containing healthy mitochondria, and next what happens is that you get a zygote. Zygote is the first cell. So this is a zygote. So this is a hybrid product. So it is containing information from the mother side and is containing information from the father side. So we get a zygote. This zygote is the first cell that is formed, and after this zygote formation, this zygote will start to divide. So initially it will form some number of cells. So it will form 7 to 8 cells. So this stage is called as your blastocyst. This stage is called as your blastocyst. So the one zygote that has been formed, it starts to divide. So in some time, in 1 or 2 day time, this blastocyst forms, and then after, from now the reference point is this. So after 2 weeks, or around 10 to 12 days or 10 to 14 days, we get what is called as your embryo. So this we call as the embryo, and this embryo is then transplanted in the womb of the mother. So let us say this is the original mother. So we'll transplant in the womb of, or the uterus of the mother. So the place where this embryo first attaches, this place is called as your placenta. The place where the embryo first attaches to uterus. So this is the uterus. And then the embryo starts to develop, and this stage we call it as fetus stage. So this is the placenta, and this is the umbilical cord. So the fetus is attached to the placenta through the umbilical cord. So this stage is around 2 weeks from the formation of the zygote, and then what? See, the in vitro fertilization gets completed till here. So till here is your IVF. So all of this thing that we are doing is done in the lab, and after that it is transplanted in the uterus. And what happens that this embryo will start to develop, and that stage we call it as fetus stage. And after the gestation period, so the offspring will be born. So after the gestation period, so there will be delivery and the child will be born. So after gestation, so there will be delivery. So this is in a nutshell the maternal spindle transfer. So we what we are doing here, we are manipulating the mother's egg cell itself. And why we are calling it as three parent baby, it is because you can see from this diagram that the zygote that is being formed here, so the it has nuclear DNA from this mother, it has so just to complete this diagram, so here you're seeing that this zygote that has been formed, it has got some element from the donor mother also. So it has got the nuclear DNA from the initiating mother, it has got mitochondrial DNA from the donor mother, and then obviously from the sperm it has got the other genetic information. So basically there are three parents involved here. That is why we refer to this as your three parent baby. So, are you all able to understand this? Shivangi, good evening. Good evening. So, we are doing the first method, that is maternal spindle transfer method. Maternal spindle transfer method. Are you all able to understand this? See, the first concept here is that you must understand that if there is defect in mitochondria of the mother, then there is a high probability that it will get passed on to the offspring or the child. The sperm cell, the father's sperm cell also has mitochondria, but the number of mitochondria is very less. So father's sperm cell, see, the cell size itself is very small, it has around 4 to 5 mitochondria only, and during the fertilization process these mitochondria are also destroyed. So that is why we say that the mitochondrial DNA is inherited only from the mother side and not from the father side. I hope this is clear. So you must understand this, that mitochondrial DNA is inherited only from the mother side. So that is why in mitochondrial replacement therapy, we are trying to replace the defective mitochondria of the original mother with the healthy mitochondria of the donor mother. So the idea is quite simple. The idea is that we'll take the healthy nucleus from the original mother and we'll take the cytoplasm which contains the healthy mitochondria, and then we'll fuse them to form a new egg cell, and then we'll go for fertilization. So we are going for IVF, in vitro fertilization, in the lab, and after the embryo has been formed, this embryo is transplanted in the mother's womb. So when it gets transplanted, it attaches to a particular place in the womb, and that place becomes your placenta. Now, so placenta is quite important for you to understand. It might help you in solving some of the questions also. Placenta acts as a barrier. It allows only the food and the nutrients and the waste to get exchanged, but it does not allow mother's blood or the child's blood or the fetus blood to interact with each other. So it acts as a barrier, it allows only the exchange of nutrients and waste. So that is why mother's blood and fetus blood does not interact with each other. Is this clear to all of you? Are you able to understand what we are doing here? Yes, I see many are online, but I hope you are able to understand what we are discussing. So this is the one of the method. This is called as the maternal spindle transfer method, because we are manipulating the mother's egg cell itself. Antigens as well, although that's a different thing. Antigens, yes, if the we'll discuss it someday. So there are some antibodies which can transfer the placental barrier, that will take some other day. Now just to make you aware that stem cells, because the next topic that we'll touch upon is therapeutic cloning. So do you know what are stem cells? What are stem cells? What are stem cells? Nowadays you would have heard that many of the parents, they are going for storing the umbilical cord, a part of the umbilical cord of their child, and they store it in some cell bank or gene bank. Why are parents storing a piece of the umbilical cord nowadays? It is not just a fashion or a fad, but there is a practical medical purpose or reason. Yes. So stem cells are popularly called as your body's master cell. Have you ever imagined that from this one cell, so we were formed as one cell as a zygote, but we are a multicellular organism, and we are multicellular, but we have so many types of cells. So you have a heart cell, you have liver cell, you have bone cell, you have hair, you have so many types, more than 200 different types of cells in our body. So how is it possible that this single cell, the zygote, is able to divide and become so many different types of cells? So the simple explanation given by scientists or biologists is that the uh, the blastocysts, they it contains a unique cell. These are called as body's master cells. These are present inside your blastocyst. These are present obviously inside the embryo. These are present in the fetus. These are present in the umbilical cord. These bodies master cell, these are also present in the fluid in which the fetus floats. So this fluid is called as the amniotic fluid. Amniotic fluid. So even in the fluid there is this these stem cells or these bodies master cells. So these master cells have the uniqueness that they can divide into different types of cells. So some of the stem cells would convert into heart, liver, or bone cells or blood cells. So that is why we have different types of cells in our body. So you would have heard about embryonic stem cells. Embryonic stem cells. So embryonic stem cells, they are present in your embryo, they are present here. So these stem cells, we refer to them as embryonic stem cells. So parents are nowadays storing the umbilical cord, a piece of the umbilical cord, because that would be containing some of the embryonic stem cells. The why they are doing it, because in future if the child uh, meets with an accident or suffers from some disease, then these stem cells could be used for his or her therapy. So using these stem cells, we can develop organs in the lab, or if there is a defective organ, so if the organ has become defective, we can give stem cells so it can cure that defective organ itself. So it has a lot of utility. That is why a lot of research is going on stem cells itself. Stem cell has the potential to solve many or to cure many of the diseases. Last year you would have come across one particular news that a woman in USA was cured of HIV using stem cell therapy only. And for many of the blood related diseases like leukemia or thalassemia, we are trying to develop some stem cell therapy. So stem cell therapy has lot of potential to solve many of the diseases. Most of the genetic diseases could be solved by stem cells, but yes, clinical trials are going on and more research are needed before we come up with commercial level therapies of stem cells. So this is the first method of maternal spindle transfer. We'll look at the second method, The second method, that is called as your pronuclear transfer method. The second method is pronuclear transfer method. So why this is called as pronuclear transfer method, we'll see. Here also the idea is that we have to replace the defective mitochondria, but there is some difference here. So let us say this is the mother's egg cell. This is the original initiating mother's egg cell. This is initiating mother's egg cell, and the problem is that her egg cell is containing defective mitochondrial DNA. These are your defective mitochondrial DNA. Now we have to get rid of these defective mitochondrial DNA, but what we do in this case is that we go for fertilization first. So we'll take an egg cell and we'll take a sperm cell and we'll fertilize it first. So this is your sperm cell and we'll fertilize it first. So we'll get to a stage where the nucleus of both this, let us say this is mother's egg cell nucleus, this, and the father's nucleus is just sitting side by side. So this is your pronuclear stage, pronuclear, and it is containing the defective mitochondria. It is containing the defective mitochondria. So what do we want to do? We want to get rid of the defective mitochondria. So this nucleus is okay, so we will extract this nucleus in nucleation. So we get rid of the cytoplasm, we extract whatever is healthy in this. So we are getting the nucleus, pronuclear here. And simultaneously what we'll do is that we'll take our donor mother's egg cell, and we do not want her nucleus, so we will remove her nucleus, the nucleus from this cell, but the cytoplasm we want. So cytoplasm is containing the healthy mitochondria, so similar step is involved. So this is containing healthy mitochondria, this is containing healthy mitochondria. So we'll remove the nucleus, we'll just take the healthy cytoplasm and we'll fuse the nucleus of this one and the cytoplasm of this one. So eventually what we are going to get is this. So this behaves like a new zygote. This behaves like a new zygote and it contains what? The healthy mitochondria. This is containing the healthy mitochondria. So now this zygote will start to divide and will form a blastocyst, and then it will form your, so I'm just using a flow diagram. So from blastocyst you get an embryo. So the process is then similar. Embryo, then we transfer it to uterus, and the fetus will develop, and after gestation period, delivery of the child. So after the gestation period, there will be delivery of the child. So in pronuclear transfer method, what we are doing is that we are removing the pronucleus, and then we are taking the cytoplasm, which is containing the healthy mitochondria, and then it behaves like a new zygote, and then we get to the blastocyst stage, then embryo, then it is transferred to the uterus. So these are the two methods of getting three parent baby. So in this case also we are taking what the nuclear DNA from this mother, this father, and this from donor mother we are taking the mitochondria. So that is why this is also a three parent baby method only. So hope this is clear to all of you. So now there are some concerns to this two methods. Okay, thank you Kino, thank you Mansa. So it has, yes, it is informative, but it has practicality also. It will be asked, or you, I'll show you the questions also what has been asked in UPSC. So let me just come up with some of the questions. Okay, so let us just try to solve this question. See what could be a mains type of question that also we'll try to discuss, but let us just try to solve this one. There is some lag. Yes, give it a try. In the context of hereditary diseases, consider the following statements. Passing on mitochondrial diseases from parent to child can be prevented by mitochondrial replacement therapy either before or after in vitro fertilization of the egg. And let us look at the second statement. A child inherits mitochondrial diseases entirely from the mother and not from the father. Yes, do you think that the first statement is correct? Passing on mitochondrial diseases from parent to child can be prevented by mitochondrial replacement therapy either before or after fertilization of the egg. Yes. So the first statement is correct, that yes, it can be done before fertilization. So in the first process that we have seen already, so in maternal spindle transfer, so we did IVF, we did the replacement before fertilization. So fertilization is happening here, so we did the replacement before fertilization. And in the second method, what we are seeing, that we are doing replacement after fertilization. So we have done fertilization first and then we have gone for your manipulation or replacement. So the statement is correct. So it is saying that passing on mitochondrial diseases from parent to child can be prevented by mitochondrial replacement therapy either before or after in vitro fertilization of the egg. So yes, this statement is correct. What about the second statement? Second statement, a child inherits mitochondrial diseases entirely from the mother and not from the father. Is this correct? Is the second statement correct? The second statement is also correct. So we have said that mitochondrial, a defective mitochondrial DNA is passed on only from the mother to her child. So there is no contribution from the father's side. So this statement is also correct. So the correct answer here is C, that both the statements are correct. Now nowadays, if you see the trend in UPSC is not going to ask you uh, only one, only two, both one and two. So they have kind of renewed the way they make the statements. So they would ask now that how out of these statements, how many statements are correct. So they would give you three statements. So they would say only one statement is correct, only two statement is correct, only three statement is correct, or none are correct. So now they have changed the way they frame the statements, because they want to that you shouldn't be utilizing the ideas of elimination. So they are not happy with elimination techniques now. Okay, so let us solve this one. This is a simple question. In the context of recent advances in human reproductive technology, pro-nuclear transfer is used for. Pro-nuclear transfer is used for. Pro-nuclear transfer is used for prevention of. So everybody is listening to the video but not trying to attempt questions. बस ज्ञान लेना है. That is okay. Okay, so prevention of mitochondrial diseases in offspring is the correct answer. So pro-nuclear transfer method. So the three in the three parent baby system, you have seen there are two methods. So sometimes they would ask you the scientific name, and sometimes they would ask you the concept which is embedded inside these terms. So the correct answer in this case is your D, prevention of mitochondrial diseases in offspring. Now here what are the some of the challenges in this? What are some of the challenges in your mitochondrial replacement therapy? So there is a religious objection. So Catholic Church, there are other medical objection, other medical concerns or challenges also, but we'll first look at this religious objection. So Catholic churches oppose one form of mitochondrial transfer called as the pro-nuclear transfer. Can you tell why? Why do you think that the Catholic Church, they are opposed to this method? Why do you think Catholic churches are opposed to pro-nuclear transfer method? Yes. So if you are aware of the debate on abortion, so you can answer this question also, that why Catholic churches are opposed to the pro-nuclear transfer method. Because the Catholic churches believe that life begins after fertilization, or when a zygote is formed, so life begins. So just because the zygote cannot speak for itself, you do not have the right to manipulate the zygote. So here the again in abortion also what we are doing is basically destroying the zygote or the embryo or the fetus. So they are opposed to this idea. So they say that this zygote formation is the beginning of life. So you shouldn't have the right to destroy life. That is why they are here. We are not destroying life. We are trying to protect life. But what they are opposed to, the idea is that you are manipulating the zygote without its consent. So that is why they are opposed to this method. See, even in this method, even in, let us say, the maternal spindle transfer, uh, whenever you are manipulating the cells, these are living cells. All cells, they come from pre-existing cells only. So even the zygote that has come, it has come from fusion of the egg cell and the sperm cell. Egg cell is also a living cell, sperm is also a living cell. So distinguishing that something is living, after fusion it is living, before fusion it was not living. So it becomes a little medically challenging, but that is the religious position. Yes, we are not destroying. Yes, the idea here, if we are not destroying, but you are manipulating. You are manipulating it. So you are manipulating it without taking its consent. Maybe when it is born, it after it achieves maturity, it would have said that you shouldn't have gone for this. So that is the argument that they are giving. So you just have to understand and appreciate their point of view. You can have your own point of view, but this is their point of view, that they are saying that you are manipulating the zygote. So they believe that when zygote is formed, life begins. So you are manipulating life after it has been formed without taking its consent. That is their opposition. So they are opposed to the pro nuclear transfer method. But let us also understand what are the medical concerns of this three parent baby. Is this a foolproof system? Is Will this lead to a permanent solution to this problem, that if the mother's mitochondria is defective, then going by these two methods, can we get a permanent cure? So the answer is no. So the problem here is, okay, let me just try to explain it from here itself. See, why it is no, it sometimes there is a problem in this method. See, when we are trying to, let us take this case. So in this case, we said that we are removing the nucleus. We are removing the nucleus from the original mother's egg cell. So when you're trying to remove the nucleus, it is not a very simple process that you are able to remove the nucleus. When you are scooping out the nucleus, so it is not like a ice cream that you scoop out the nucleus and along you get only the nucleus and nothing from the cytoplasm. So when you're scooping out, let me use a different color so that it doesn't confuse you. So when you're scooping out the nucleus, so it might happen that some of the defective mitochondria from the cytoplasm is also carried. So this nucleus might be having and during this enucleation process, you might carry some of the defective mitochondria also. If you are carrying some of the defective mitochondria, it means in this zygote also you will have very few defective mitochondria also. So yes, 99% of the mitochondria in this new egg cell that you're seeing is healthy, but let us say 1% is defective. This is like 1%. 1% of the mitochondria is defective because from the enucleation process you have carried forward some of the defective mitochondria, and in this new egg cell what you are seeing that most of the mitochondria is healthy. But even if there is a small percentage of unhealthy mitochondria, what has been observed in studies is that what happens eventually, that this defective mitochondria, the percentage of this defective mitochondria starts to increase and the percentage of the healthy mitochondria starts to decrease. So by the time the child is born, you could see the reversion itself, that the defective mitochondria becomes, let us say, around 40%, and the healthy mitochondria in the child is now remaining only 60%. So this is what is referred as a reversion, or the defective mitochondria is relapsing or it is multiplying. So this is one of the medical concerns in your three parent baby. This can happen in both the scenarios. It can happen in maternal spindle transfer also, it can happen in pronuclear transfer also. So when you're trying to remove this nucleus, you might carry some of the defective mitochondria along with this in this enucleation process. So it might have some defective mitochondria also. So this reversion is a concern. This is a medical concern. So there are basically two concerns. One is obviously the religious concern, and because of that there are other ethical concerns also. So when this three parent baby was legalized in UK, so there was also a debate that sometime in future we can misuse this technology. We can misuse the technology to produce what is famously referred to as designer babies. So maybe in future, here we are doing manipulation. We are going for mitochondrial replacement therapy for altruistic purpose. That is, we want that the child should not get some disease. That is an altruistic purpose or vision. but in future we could misuse this technology and we can get babies which we desire. So this could lead to designer babies also. But the opponent of this point of view, they say that we are just manipulating the mitochondria. We are just manipulating the mitochondria. We are not manipulating the nuclear DNA. So to get a designer baby, let us say you want to have a designer baby who has a blue color eye, or have a particular height, or anything that you desire as a genetic trait. So the opponents to this idea, they are saying that we are not manipulating the nuclear DNA, not manipulating the nuclear DNA. So most of our genetic characteristics, that is how do we look, how do we appear, some of your other characteristics, they are determined mainly by your nuclear DNA, and very less information is there in your mitochondrial DNA. So we're not manipulating the nuclear DNA, we are just manipulating the we are just manipulating the mitochondrial DNA. So there is no concern of designer babies. So that argument is nullified. But sometime later we'll also, or you would have also heard about this Chinese scientist, He Jiankai. He had produced world's first he had produced world's first gene edited babies. So he had manipulated the nuclear DNA itself, gene edited babies, and these babies, they are said that they are resistant to HIV. So this was a news of 2018. So this argument that that such manipulative techniques can also lead to designer babies still holds the ground, that some scientists have started manipulating even the nuclear DNA also. So that is a concern in this case also, or in future also. So in India it is not legal, so we are not going for three parent baby. So USA also, it is not legal. It is only legal in two countries as of now. It is legal in UK, and last year it was made legal in Australia. So these two countries have made it legal. What can you expect in mains? So in mains they could frame a question like this, that what do you understand by three parent baby? They could ask a simple question like, what do you understand by three parent baby? What are the ethical concerns related to three parent baby? So, or they could ask in something on these lines, that the concerns outweigh the benefits. Comment in a three parent baby. So that could be a line of questioning. So you know the technique, now you know some of the concerns or challenges also, so you can frame an answer based on this. Is it okay? So therapeutic cloning. Okay, so therapeutic cloning, it would take some time, but I'll just introduce that topic so that sometime later we'll discuss this also. What do you understand by this term? Let me just discuss a broad term, cloning, then we'll look into a specific type of cloning. What do you understand by the term cloning? So now we are moving to the second topic. We'll briefly discuss. So maybe I'll take another session and I'll explain this in more detail. But what do you understand by cloning? What is a clone? क्लोन, नेचर भी क्लोनिंग करता है. कहाँ पे आपने देखा है क्लोन? Have you heard about twins, identical twins? Yes. So similar genetic composition. So identical twins, nature does cloning. So that is a natural method of cloning. Uh, in when we are talking about cloning here, yes, bacteria is also. So any organism which goes for asexual method of reproduction. So these methods of reproduction that you are seeing is called as sexual method of reproduction. Why? Because we are taking the genetic material from two genders and then it is being combined in the offspring. So this is a sexual method of reproduction. So what you are getting here, this is not a clone. So you are not a clone of either your father, neither of your mother. You have genetic composition from both your parents. So you are basically a hybrid. You are not a clone. You are hybrid of your parents. But when you say about bacteria, so in bacteria they do not go for sexual method of reproduction, they go for asexual method of reproduction. So they go for, they basically they form clones of themselves. So when we talk about cloning at this, we are talking about artificial cloning. So in artificial cloning also there are three types of cloning. So one type of cloning is called as your reproductive cloning. Another cloning is your therapeutic cloning, therapeutic cloning, and the other type of cloning is called as gene cloning. See, all of you must have gone through RT-PCR test, RT-PCR test to test for COVID. So when you're going for PCR test, or this we are trying to do what, we are trying to make multiple copies of the DNA. So when we are making multiple copies of the same DNA, that is also cloning only. So that is called as your gene cloning. So what is reproductive cloning and what is therapeutic cloning? So we'll discuss this in detail maybe next time when we schedule it. So just to give you an idea what is reproductive cloning, do you have any idea that what was the first organism that was cloned? The first organism that was cloned using this method was Dolly the sheep. So she was the first reproductively cloned animal. Therapeutic cloning involves a particular concept that we'll see sometime later. If you have any doubt with respect to the mitochondrial replacement therapy, you can ask. So let us open the house for some discussion. Because this explaining therapeutic cloning will take me some time. Maybe I'll schedule another video and that will do this thing in detail. Yes, any doubts? Okay, if you do not have a doubt, then I'll ask a question. Okay, what do you know about germ cells and somatic cells? What are germ cells and what are somatic cells? Yes. What are germ cells and what are somatic cells? So in order to understand cloning, we have to understand the different types of cells in humans or in animals basically. So yes, Mini, quite correct. So somatic cells are your body cells. What are germ cells then? What are germ cells? Germ cells are the cells which are present inside our reproductive organs. So those are also called as your sex cells. So germ cells are also referred as your sex cells which are present inside our reproductive organs. And all the cells other than the germ cells in our body is called as somatic cells, or they are called as body cells. So most of the cells in our body are somatic cells only. So your heart, lung, liver, skin, bone, so they are your somatic cells only. So we have germ cells inside our reproductive organs. So when we are trying to make clones, so what we have to do, we have to make clone of the somatic cell itself. So we have to make multiple copies of the somatic cells. Another simple question to all of you: so do you think that the genetic information in your, let us say, heart cell or your liver cell or your lung cell or in your bone cell, the genetic information, if you open the cells of this and if you look inside the nucleus of this, do all these cells have the same genetic information or do they have different genetic information? Okay, so try to find the answer to this, and we'll take this reproductive cloning some other day. So hope to see you again. So I'll close the session if nobody else has a doubt. So if anybody has a doubt, you can ask.